Are you gonna start, Ella?
Okay.
I am.
Awesome. Whenever you're ready.
Check through that it's piping through to Twitter. Yes, it is. Cool.
Amazing.
Hello, welcome to everybody watching. Today we are chatting to Dimitri Scherbakov and Rashid Akbergenov, who are the researchers behind Project Transfidelity. We're going to be talking about their research and giving a little announcement. And also, it's a very collaborative space, so please also remember if you want to ask any questions, pop them into the chat. But yeah, we're really excited about what's happening with Catalyst. We're very proud of what we're doing. And yeah, I'll hand it over to Benji.
So yeah, the Project Transfidelity, which is led by the two researchers here, we spoke to them, I think for the first time a few months back, decided that, they wanted to fundraise for their project via Catalyst. We then kicked off the project, I think about 29 or 30 days ago. And as of today, the project got successfully funded. So I think this is the first time you guys have heard this, but congrats. Like your project was successfully fundraised. So big ups to you guys. Just wanted to share that news with you.
Completely or almost completely?
Completely. Project is completely funded. Yeah.
We are proud of ourselves, yes. And of course of you. Yeah.
No, all the credit goes to you guys. Really, really interesting project that the community was clearly very excited about. Very big shout out to some of the DAOs that contributed to it, including Vita DAO, Cerebrum DAO, as well as Beaker DAO. So all three of those communities decided to contribute substantially large amounts, and we also had enough individual contributions to get the entire thing funded. So yeah, huge props to you. Big thanks to everyone who contributed. And we're really excited to get the project kicked off, or I guess continuing, because you guys have done a lot of the great upfront research as well. We're also gonna add Michael Torres, who I think is here. So Michael Torres.
Hi, Michael. Yeah, so nice to see you.
Yeah, nice to meet you.
Nice to meet you guys. Actually, I know Benji, you might give me an intro, but I was going to say, so I've done a few companies, including through Vita DAO and IP Molecule platform. But more importantly, I've done work on nonsense mutations and read through and other things. So seeing,
Wow. So you're familiar with the stuff. You know what means genetically, yes.
Yeah. So I'm actually really excited. And one of the projects that I'm working on with Vita DAO is to suppress arginine nonsense mutations, where arginine, occurs in many proteins involved in like DNA damage, DNA repair. If you look at arginine, and particularly ones that have the CGA codon, which cytosine is, can be mutated very easily. Like that's our approach. We think if we can keep protein levels up that have arginine, that gets downregulated over time, that could also affect lifespan. So very cool to see this project.
Okay.
Get on.
Yeah, hitting the same button. Yeah. Okay.
Yeah. Cool.
Yeah. And in fact, as of today, you guys are going to be joining even a smaller club of people who've been funded by the DeSci community. So that's pretty exciting as well.
Yeah, congrats. Then you can get to work, so.
Yeah. I'm patiently expecting this moment when we can start finally doing these things. Yeah.
Very cool. Yeah.
Also, we're about to add to the stage Brian Magursky, who is the lead at Cerebrum DAO. Hey, Brian.
Hi, everybody.
Hi, Brian.
Anyway, maybe quickly go through intros here. Brian, do you want to introduce yourself, and then Michael, go for it, and then the researchers can as well.
Yeah, absolutely. So I'm Brian Magursky. Nice to meet everybody here, and to everybody joining as well. I'm the founding steward and strategy partner over at Cerebrum DAO. I founded this network organization to fund accelerating solutions and therapeutics for neurodegenerations, and with a very specific focus on Alzheimer's as well. close to a little bit over a year ago, been really thinking through how to change the game in neurodegeneration and Alzheimer's research for quite some time, last several years. Really tied to a mission that got launched with my youngest daughter, who now is 13, but she was born with Down syndrome. And my awareness when she was born of the population of people with Down syndrome have a guarantee of early onset Alzheimer's based dementia, and really wanted to focus on this. How do we accelerate solutions and therapeutics, just given that the past 30 years really hasn't seen a tremendous amount of progress in at least in terms of impact outcomes for humans in the entire field of neurodegeneration, but specifically in Alzheimer's, despite large increases in funding. My background prior to this was all software. I've been a software entrepreneur since about 1998, starting several business-to-business software companies. So I don't come at this from a biology or a neuroscience background. I come at it from just an entrepreneurship and technology. And one of the ways I thought I can help make a difference here is leveraging technologies like distributed ledger technologies and blockchains, and community growth and fundraising mechanisms to help empower the people that are doing the research and drug development for these cures, such as the researchers for Project Transfidelity here. So we seek to be an enabler, and that's what I was hoping I can contribute to this field.
Amazing. And do you mind briefly just mentioning what got you so excited, particularly about this project, and made you excited to kind of share with the Cerebrum community?
Yeah, I see Marina just joined. Marina's our lead for our neuroscience and translation group. And she can probably answer that question even better than I can. But we have senior reviewers and a really good neuroscience and translational drug development set of expertise. And I think the credibility of the project, the target, and the researchers and the approach that they're taking got us very excited that this could be a potential disruptive opportunity in the field for Alzheimer's. So, Marina, if you wanted to say anything specific on the science, I'll let you jump in right there.
Yeah, I think that we were looking at several projects in the recent months. Some were more advanced, some were less advanced. This project is definitely early in the translational pipeline, but what we know from other organizations, in order to change something in the field, you really need to make bets on different areas. And that was the reason why actually all our senior reviewers agreed that we would like to participate in this project. So we are now very early, looking forward to the next year and the results that you get with the research. And then, well, if the hypothesis that you have thought will be confirmed, we will be happy to support you further and advance this project to the next level.
Yeah, I'd echo that thought. Our excitement about this at this earlier stage is, and this is where we think an organization like Cerebrum can play a very value-added role. So we intend to be very collaborative in this, in advising on the research setup for translation, and so we're looking to partner, and if we can see promise in this, we've even discussed that we would be very interested in leading a follow-on round for this as well, if we see the promising results in the first phase.
Great. Yeah, thank you very much for your trust and your effort for this, to support us. We of course we cannot guarantee the success, right? But what we can promise, we will do whatever we can. And it will not be like we just kind of raise money and then we just sit on it. We will really use it to maximum, let's say, possibility to really accomplish our goal. So I mean, this is what we can promise.
Awesome. Michael, want to say a few words? I know, you're kind of the opinion that many of these age-related diseases are caused by transcription errors and nonsense mutations, but what gets you excited about the other side of RNA polymerase?
Yeah, so I'll just do a quick background. So for those who don't know me, I'm Michael Torres, scientist by training, but co-founded a company called Recode Therapeutics, which is a genetic medicines company that's now raised over 400 million. It was my postdoc project in the lab. And then I took it through an 80 million Series A as the, one of 3 employees. And then we, continued to grow. And the program that I worked on, cystic fibrosis is now in the clinic. And then after that, focused on an ADC company. So I'm the CEO of an ADC, antibody drug conjugate company that's based here in Houston, where I'm at. That's currently closing on a $14 million seed round. And then have a longevity project that I took through VitaDAO that was funded through VitaDAO, and then was recently tokenized as the IP NFT this year called Vita RNA. And so we raised about $300,000 to build a gene therapy vector that focuses on addressing, as Benji said, another side of aging, but in a very similar sort of framework. And so I'm a big sort of believer, and in fact, some of their data showing with the aminoglycosides, and if you mess up translation, that's, I think for those of us who have a science background, we understand that's generally bad, but just sort of the downstream consequences to all cellular processes, I think was actually, is quite strong and quite evident, and I'm a big believer in that. So conversely, on the other side, right, to be able to, maintain fidelity of your proteins, and by then, cellular processes for longer, autophagy, every kind of thing that's like associated with aging and cleaning up junk, it's not hard to imagine that could be, positive for lifespan and health span for us. So I was really intrigued to see, inklings of data, and perhaps there's a lot more that I should just read in the literature myself that shows that you can find molecules that can help increase translation fidelity, and if you can sort of take those hits, understand them, understand what processes that they're going after, but more importantly, as a drug developer, understand the potential liabilities of these drugs. This could be a whole new sort of class of medicines that could have wide-ranging effects, and so, like, again, just to point to it explicitly, like I'm certainly in the camp of you're going to need like there's multiple aspects of aging, and the drugs that can cover as many cellular processes or be pleiotropic, in other words, like have multiple effects across those processes, are the ones that are likely to have a significant benefit that we can actually see and capture and measure in humans. And so I'm really excited to see where this project goes, and I certainly was an early supporter from just getting it to this point, and then happy to see it through Catalyst as well. So cool.
Perfect. Thank you.
Sure. Yep.
So Benji, how should we proceed?
Yeah, no, would love to hear from both you and Rashid. I think a few points might be useful to touch on. I think number one, maybe recapping where you are with the research. And then also, now that it's funded, where is this going? The money that has just been raised, what is it going toward? What data do you hope to generate? And, in an ideal case, how would the research proceed?
Okay, so that means that we should skip introduction from our side, where we're from and what we are doing, and so on and so on.
You can do that too if you want.
Yeah, so then very quickly. we are both from the basic science. We are specialized in translation. We studied translation for more than 30 years now, I think. And last maybe 15 to 20 years, we spent studying the inhibition of translation, so namely development of new antibiotics. So it's aminoglycoside group. And during this study, we found out, so it was actually known that those antibiotics causing the mistranslation. So they're not only block ribosomes, but they also made them less accurate. So the bacteria has two problems simultaneously. So it's less proteins, and the proteins which they still produce, they are wrong. And that's why probably those antibiotics are actually bactericide. And then we decided to check what happens with the higher eukaryotes if we decrease the translation fidelity. For that, we used a genetic modification approach, and we could find out that in a mouse model, those decreased fidelity leads to premature aging and also development of certain neurological disorders which resembles the early stage of Alzheimer. As we all know, there are no Alzheimer or Alzheimer-related disease in mice, but we could find that some neurological disorders they expose, they really resemble the early stage of Alzheimer. We could also find that they have increased, among of other phenotypic changes, they have increased protein aggregation in the brain. And that all leads us to the conclusion that maybe we should try to do opposite, namely to increase fidelity of translation and check whether it will be beneficial for the lifespan and also for the neurological health. Unfortunately, when we would try to do it in mice, just like basic research, normally, it will take enormous time. So it's clear that it will be at least 5 years project, maybe more. And therefore, when we really start to plan it, we decided to cut this this long way and to try to do it with compounds, because we all know that there are compounds which decrease translation fidelity, it's namely aminoglycosides. Why not try to search the compounds which increase fidelity of translation? I can explain you from structural point of view how it should look like and how it should work at the level of ribosome, but actually it's just, trust me, it is possible. So just structurally it is possible at the level of ribosome. To our surprise, we have found that our lead compounds, they do not work probably at the level of the whole ribosome, of the ribosome directly, but they probably have some indirect effect. And yeah, so then now we come more or less to the questions which you send us, the list of questions from fellows. The question is, should we follow them, or what is our
No,
plan?
don't worry, don't worry. I actually, so I wanted to also ask you guys, I know you both have sort of over 25 years of experience in the lab, and have been working together for 15 years. So I'd love to discuss like how this project came together with both of your skill sets and like what made both of you decide to do this together.
Maybe I can say. Actually, we know each other now probably for 17 years, yeah, roughly 17 years. So we worked together in the same lab for 15 years. And interesting, we had some common like friends and places which we worked and studied and something. So we had also like we are also by age at the same age, and education-wise also very similar. So we very easily got together, and but we had let's different kind of strong sides from both of us. Dimitri is very systemic, and I can be like innovative, something like this. And in addition, he has many let's say other skills, and and maybe some of the skills which I also have, which he doesn't have, and something like this. So we, in general, we combine very well, let's say, and we realize that when we work together, we're really very efficient. So like we took several projects, and all of these projects were quite successful during this 15 years collaboration. So, and then very often, of course, we had some common coffee or whatever. And as soon our research concerning this protesters research, I mean, studying ribosomal fidelity, and we also worked in different aspects, as Dimitri mentioned, about the drug development, but this was actually purpose was to discover new antibiotics against bacteria. We thought there should be another application for our knowledge. We realized that this fidelity of translation is very important, and it is very important for aging, very important for, it's somehow related with age-related diseases. And then this idea, it's practically was on surface. We start to discuss what could we do about this, and actually, finally, we came up with the idea, maybe we should try to do some applied stuff. We accumulated enough knowledge, which could be used for better purpose, let's say, to make it like useful for more people. And it also was for us to see whether it's really it can be applied and really cure some people using our knowledge. And that was actually it, how it came to our mind. So that's probably too long.
No, that was great. Thank you so much.
Yeah.
So also, what I am going to do also, if everyone's okay with it, is like for our viewers that maybe aren't super familiar with this project is called Project Transfidelity, and Dimitri and Rashid have been trying or have found 2 compounds that they suspect improves the accuracy of the proteins printed in our body. And so sort of the funding used for this project is going to be to learn a little bit more about those 2 compounds, to test them a bit better, to understand how they're acting in the cell. So sort of that's the overview of what the funding raised on Catalyst is going to do. If either of you want to explain a little bit more about what the sort of experiments are that are going to happen with this funding, and sort of really just talking about what has been enabled by this funding.
Okay, so probably I can answer. As I already mentioned, we have compounds which have indirect mode of action, which means that those compounds do not interact directly with the ribosomes because they are not active in vitro system. That was one of the questions actually. What are the difference between 2 systems we are using for screening? And that's exactly the point. So we have cell-based system where you have a membrane and you have a whole cell and you have the whole processes which are going inside. So there the compound should penetrate the cell first, but then it can either directly or indirectly affect translation. Another system is in vitro system where you have clean ribosomes and translation mix, and there only the compounds which directly interact with translation apparatus will have effect. And we actually expected to find probably more compounds which will affect translation directly. But to our surprise, we have found that the most interesting and promising compound we found till now were with indirect effect. It means that they improve fidelity of translation. We don't know exactly how. We have some gut feelings, and we have the preliminary hypothesis, which is based on the publication done on yeast, that under certain conditions, yeast can improve fidelity of translation, namely when you put them on a hungry stage. They try to modify ribosomal proteins so that the translation slightly goes down and simultaneously the fidelity increases. It's hydroxylation of one of the ribosomal proteins. That's what we expect actually to have to take place more or less in our case as well. But what is the pathway going to that target? We do not know. And that's one of the major tasks we want to accomplish using the funds you provide, actually. If we would be able to identify the pathway, normally you have several targets along the pathway, and we can not only think about improvement of our compounds, but we can also think about the other targets which can be affected maybe even more effectively within this pathway, so that we can improve fidelity even more. That's major task. Then, of course, we want to work on improvement of the compound just chemically. So we plan to discuss it with organic chemists. We have one in our team. This is Professor from Basel, Gillingham from Basel. But maybe we will be invite somebody else, because it depends on the specific specific molecule we want to modify. Then, of course, we want to accomplish a small but important task. We need a control. We want to produce the cells which are genetically modified, so that the translation fidelity is improved, and check what happens there. Because this system is bona fide, in contrast to our compounds. It is described, it is published, that it is possible to improve translation fidelity with genetic modification, and it has beneficial effects, at least on lifespan, on flies and worms. Unfortunately, people who published it, they do not work anymore on this topic, as far as I know. And second, they did not check aggregation of the proteins, unfortunately. It's, of course, very difficult to do it in worms or flies, but they also produce cell culture, and they didn't check it. Sorry. That's what we plan to do. Because for the moment, it's very difficult for us to estimate quantitatively what happens. We know that how much our compounds improve fidelity. We can estimate the level of aggregation, which is decreased with this level of translation fidelity improvement. But we have no idea of what is theoretically possible until where we can go. And there, the bona fide restrictive mutations, which are described for bacterial and now for the eukaryotic organisms as well, it's a great benefit for us.
Amazing. And so also,
actually, there is one more point.
Yeah.
I should probably mention. This is we have to check these two leading compounds in IPS system. Yeah, because let's say cell culture where we have like stem cells which were like developed to neural cell, and then we have already different approaches to measure accumulation of aggregates in these cells. So in presence of our compound, we should demonstrate that aggregation is decreased. So
and
this is actually one of the important points for our proof of concept. So
and
covered.
The induced pluripotent stem cells, those are human derived, correct?
Absolutely, yeah.
Yeah. So I think what that also is helpful in showing is sort of how these compounds would interact in human cells, presumably.
That's not even only human derived, they are actually patient derived. So it's really disease related IPS derived neurons. For the moment, we have access only for the Parkinson neurons. But there we should see that also to address one of the following questions. Actually, according to our expected mode of action of our compounds, it should be beneficial for all types of neurodegenerative diseases which are aggregation, so protein aggregation related. It means Alzheimer, Parkinson, Huntington, frontotemporal dementia, Lewy body dementia, and some others which are less well known. And therefore, for the moment, we have access to the Parkinson derived neurons, IPS neurons, which is good. And what is also important, it's rather easy to detect the positive effect. Because in contrast to the existing Alzheimer models, in Parkinson model, you can very easily detect the aggregates of alpha-synuclein in contrast to non-aggregated alpha-synuclein, and this is, of course, beneficial.
Amazing, thank you. I think one of the sort of important things about research is like kind of having a forward-looking approach. So I'd be really curious to know like where you hope to see this project go over the next couple of years, and like what the next sort of important steps would be.
Next steps would be probably if we identify, as Dmitri already mentioned, if we identify the target. So it means we will identify pathway, and then, as he also already said, we can modify existing compounds to make it more efficient and having less side effects. But also we can already consider other targets like downstream of the pathway. We can find some good molecules, like whatever it will be, most likely some kinases or some acetylases, whatever. And this enzyme should have some possibility to be targeted, depending what it should be, either inhibition or induction of the function. And it means now there are plenty of different approaches to design like small molecules fitting like key to the lock to the different enzyme. And for example, in Basel, we have special service even working there, and they develop different types of small molecules fitting to different targets. So if we have targets, there are plenty of possibilities to go. I mean, different ways to go. This is one thing. Second, will be like we may, in addition to this, we may search for other compounds, like to do screening. As you know, we have developed these two platforms for in vitro and cell-based screening. And I think if we have enough capacity, we can continue screening, and maybe we will find even better compounds, which will be targeted some different targets. I mean, and maybe we will achieve even better increase in fidelity. That's different as a way to go. Theoretically, we also considered another option like genetic approach, like, but it's of course long-term plan. But theoretically, it's possible to identify new mutations in ribosomal proteins and introduce this mutation at least in some model organism to see and demonstrate that this may solve these age-related problems as well. So, and in future, finally, I think it will be allowed to do some gene therapy, and we will be ready for this. That's what I think the future plans.
I would love to know why you guys were open to sort of fundraising on Catalyst. Obviously, you know, what we do is quite a novel fundraising method. So yeah, why were you open to it? What has your experience been with funding on Catalyst and with the DeSci community been?
So actually, it's a little bit difficult questions because we were somehow introduced to you by Apollo company. So, and then our first experience with the Catalyst and with the molecular team was so, how to say, pleasant that we just decided to stay with this possibility. And yeah, so it's just wonderful work, I must say. Yeah, so it's rather difficult, rather different from the what I heard about fundraising on the courses, which I how to say did for the biotech entrepreneurship. So it was rather easy, so straight going, and yeah, it's just nice possibility for us. which is also important for what is also important for us. It's a non-dilutive finding for the moment, and we are happy to share our IPs, because actually even not we share IPs, but our university shares IP with you, and we just have money for our research. That's our goal, and this is a wonderful win-win system. So what can I say? Nothing could be better. Just maybe to find Emilia on the street.
We'll keep looking. Amazing. Also, I think what, like in my experiences, sort of having spoken to you guys before, is how sort of open you are to like this collaborative approach, where it's basically, you know, everyone comes with their own set of expertise, and I think, at least, you know, from our end, that's been something that is really important in decentralized science, is this ability for everyone to group together and use this hive mind, as Benji always calls it. So yeah, I was wondering what your opinion on that was.
That actually, even during preparation of the project, the project itself was like developing, and I myself was surprised that actually we worked on this project for many years before, but when we contacted with you people, like with it was like observed from certain point of view which is completely independent and I so-called naive point of view, yeah, and then it make us clear and our eyes were open because of this contact. So I think when you rearranged our let's say plan, prepared the new presentations, this all actually fit so well like piece of puzzles together. And I see now that the project is crystal clear, straightforward, and every step is coming from the previous one. It's very logic, and I think we will within this time of frame of this project we will have clear answer. Does idea work or not? So this is I think the most important question we have now.
Yeah, I'd jump in there. You know, what is interesting about this project in particular, even different than the other projects that have been funded in decentralized science so far by singular DAOs, is what you have coming together here with the funding is support from at least 3 different DAOs that I know of. So Cerebrum, Vita, and Beaker. And then a bunch of individuals that are just personally interested in this particular space. So what you're getting with that at this early stage is you've got at least 3 communities with motivated participants from different aspects. So one with Vita from a longevity perspective and just trying to find ways to slow down the aging process. With Cerebrum, we're definitely focused on the neuroscience and neurodegeneration, but we also take a grassroots approach just to say that let's look at these diseases from a perspective of the aging brain and the aging process and figure out what's going on to help slow down and eventually prevent. And so each of our communities is filled with experts as well as just individual patients that are trying to have solutions to you know, these problem spaces that want to be engaged in the research. And then with Beaker, you have a unique community that is filled with people that have put money into advancing this new decentralized science way of funding and accelerating scientific solutions to human health problems. And that's a community of people that want to see these mechanisms like IP tokens and marketplaces like Catalyst succeed, grow, and thrive, and continue to invest in that. So, so you really this is really powerful when you take the research that you guys are doing and the challenges that are in front of us together now because we're partnered. And then you bring these communities that are all motivated to help and participate. We've seen this with rare disease communities when they pull their resources together to try to solve a specific problem that pharma isn't solving for them. And it's that hive mind, or it's that group of people that are highly motivated and aligned in interest that help break through the set of challenges. And so with this project, it's really exciting to see you're getting the power of a few communities coming together to add and to be a force multiplier for the work that you're doing on the research side to help it find its way to successful outcomes, which is ultimately going to be, solutions in humans, right?
So I can add to all of that actually we have already one piece of very positive effect of this common knowledge, let's say, or collective common sense, even I can call it, because during our last session with Bika, we get somehow not completely unexpectedly the very nice new idea, and actually it was so very basic, let's say, but just basic knowledge, just common sense, but somehow it did not came in our mind before. So we're really happy to hear that. And namely, somebody asked us whether we tried to mix those compounds. And we said, wow, no. And it could potentially, if it will work, it could potentially solve even two problems simultaneously. First, it could be at least same effective or even more effective. And second, it could lead to going out of the protection, so patent protection. If we can establish the certain ratio of mixing, which works the best, then we are so new with this mixture, and that will be something really so new, and we can probably even patent it as a mixture. Yeah. So therefore, I think this so common sense and collective work, collective brain work, is something really very attractive and very powerful.
I could not agree more. I think on that note, Benji, would you like to explain sort of what maybe the next steps will look like for this project?
Absolutely. So we are currently in contact with the tech transfer office at, I think it's called Unitectra, which is affiliated with University of Zurich. They will handle kind of the licensing agreement. We're in the final process of finalizing that agreement. We'll keep the community, as posted as we can. However quickly we get information, we'll disseminate it. But yeah, the goal is to finalize that IP agreement as soon as possible. We are currently working on some smart contract upgrades so that we don't need to wait for the agreement to be finalized to launch the IP token. So we may be able to actually launch the IP token prior to finalizing the agreement. This isn't confirmed, but we're currently in the midst of building / discussing this. But yeah, suffice it to say, we should have an IP token trading on the open market as early as next week. And then, yeah, we'll accelerate the conversations with the technology transfer office as soon as possible. And then after which the funds can be transferred and the funded research can begin. So yeah, I mean, like we're moving things as fast as we possibly can on the university side, on the technology side, and really we're just excited to start generating some valuable data on these compounds, how they work, how we can modify them to make them work even better, what combinations of them can potentially create synergistic effects. We're really excited to actually get down into the data. So yeah, that's kind of it as an update from the technology transfer office side and from the token perspective. But yeah, I guess Dimitri and Rashid, as soon as we can get the funds wired over to the university, when is the first time you think you can generate data? When should we expect for some sort of scientific update?
Oh, Dimitri, you're on mute. I can see.
as soon as you want, let's say. So
No, there are milestones. Yeah, we have
yeah,
time.
sure.
Like we generate it now. Yeah, I want it now. You
No,
generate.
I mean that the real gentleman always has something to say, you know.
We
So
we want
He wants
we
to
want
say
to
that we have something already. We didn't sit just waiting for crowdfunding. Yeah, we continue to work even in parallel. Yeah, and there are some promising things already going on. We didn't want to uncover it because it could be false positive result. That's why we don't want to be like suddenly giving some promise and then not keeping it. But we know what to do. So it's let's say one of the milestone is IPS, right? So this IPS stuff can be done how long? It's 1 month, right? Shortest 2 months.
I think in milestone it's written till the end of this year.
Mm-hmm.
Correct me if I'm wrong.
Yeah. So
But yeah, so preliminary result we can demonstrate earlier.
yeah.
That's no question.
Yeah. Then PISA, it's if we are lucky, it can be, let's say, done in parallel, of course. And this is fastest maybe 1 month, but let's say to be sure, I would say it's 2 months time, and then we have, let's say, already probably some targets, good candidates for targets. But again, for targets, we need verification, right? So it means you have to knock it down in order to see that effect which was there because of this drug is not anymore because you knock down the target. So it also will take some time, just this kind of experiment. It's not enough just to find something, considering it is a target. You have to prove afterwards that it is really the target. So you have to challenge your findings in order to be sure that your finding is correct one. So it's you understand, right? So it means first results will not be earlier than, let's say, 2 months.
Okay,
Yeah.
totally understood. Well,
Yeah.
as you're conducting the experiments, if you want to send updates to the Telegram group, we'd love to see them. If you want to put a body cam on and stream yourselves, pipetting, I'm sure the community would love to see it. So yeah, keep us posted. There's a lot of people eagerly awaiting your results, and kind of going back to Brian's point about you now having access to a large decentralized community that is emotionally invested in this project. We're all very excited to see the data that you generate, and we're here to support you as much as needed. So as much as you need help designing experiments or interpreting data, we'd love to help. I'm sure the community would love to help too. So let us know how we can support you, and keep us posted, and we'll add you to the Telegram group so that you can have a way to communicate with the entire community.
Very
perfect,
well.
perfect. Thank you.
Great.
Cool. And also just a reminder to anyone who's watching this live stream, you can also join the Telegram group. There's a link for it on our Twitter. So come on over. We can all chat science together. Awesome. Are there any open questions? Anything anyone's burning to say? But other than that, I think we can wrap it up. I know it's coming on dinner time soon.
So we will probably clarify further the practical part of the money transfer and so on, because it's a technical stuff, how it should be done. So
100%. We'll be in contact with you. Yeah.
yes.
All of the technical stuff will happen.
Mhm.
This was just to let you guys know and to celebrate a little bit and say just like a massive well done to the both of you, and a big thank you as well for choosing to fundraise with Catalyst.
So thank you, thank all of you, and we are happy, and we will try our best to fulfill what is expected from us.
We know you will, and science is hard, so
Yeah, we all
wish you
know
the
that,
best
right? But you have some fantastic partners with you now, so we're looking forward to this.
I just can say that it was really great pleasure to work with you. I enjoyed it a lot. It was very efficient, very effective, very fast, and precise. So I hope we continue, and I hope we will succeed. Yeah.
And last but not least, you did such a nice presentation. I am still under impression of that. Because all our former presentations were somehow, like normal scientific presentations without, anything which is pleasant for the eyes. But now it's just piece of, I don't know, piece of art.
We'll pass along the gratitude for our designers. But yeah, I mean, you guys, you can't have it all. You can be good at science, or you can be good at making presentations, but you can't. We're not going to let you be good at both.
So yeah, unfortunately, that's true. Yeah.
But anyway, hopefully this will let you focus more on the research, which is the actually important, very, very difficult aspect of it, and we can handle everything else. But yeah, thank you.
Very good point. Yeah,
thank you for your trust in us. It's a pleasure to put our trust in you guys, and we're really excited to see what comes out of your project.
Thank you.
Thank you very much.
Cool.
For your trust.
Okay.
So,
bye, guys.
Bye
bye,
bye.
everybody. Thank you.
See you next time. Bye bye.
Bye for now. Yes.